Hi everyone, I'd like to present you what I have done during the project called Visualization Tool for Olfactory Bulb Data during summer of HPC program at Cineca in Italy. Olfactory bulb is a structure in brain that is a part of olfactory system. It takes odor input from olfactory receptor neurons, process and transmits them into the other parts of brain for additional processing. It is believed that olfactory bulb has an important role in odor recognition. One of the most significant neurons located in the olfactory bulb is mitral cells. When mitral cells have odor input, they create unique pattern for each type of odor input. Similar smells produce similar patterns. These patterns become more unique and more easily identified as the time goes on. With the help of patterns of the firing neurons, how odor recognition works in olfactory bulb, especially in mitral cells, is tried to be understood. The motivation of the project is to develop a tool visualizing olfactory bulb neural network to help researchers in their studies of odor discrimination. Mitral cells in olfactory bulb get information from olfactory receptor neurons, process the input, and transmit it to the granule cells. A mitral cell is composed of several underlying structures such as tuft, epic, soma, and dendrites. Tufts of a mitral cell get electrical signal from other neurons. The electrical signal passes through tuft, epic, soma, and dendrites respectively. The number of dendrites is much higher than the other parts of a mitral cell. Dendrites transmits the input to the corresponding granule cells. Our olfactory bulb neural network consists of 635 mitral cells. The neural network designed in neuron software, which is a neural network simulator. For the scientific visualization, Blender software is used. 635 mitral cells consist of approximately 400,000 segments in total. To render the structure, two different rendering approaches are implemented and they are used concurrently. Rendering the whole structure currently takes less than 5 minutes in Blender. To identify the patterns, when input, in other words, electrical signal, arise in a mitral cell, segments of the mitral cell are colored to visualize the propagation of the input. The colors represent the strength of electrical signal passing through a segment. Purple segments represent the segments that have no electrical signal. The strength of electrical signal is increasing through purple, dark pink, red, orange, yellow, and white respectively. White segments show the center of currently moving electrical signal. The simulation time is approximately 40 seconds. In order to show the electrical propagation, 300,000 frames are used in total, which makes the animation duration around 200 minutes. This part of the video animates a small part of the whole simulation. Dendrites are surrounding the surface of the structure and make the surface spherical. The electrical signal coming to dendrites goes to granule cells, which are not available in the scene. There is no signal transmission between dendrites of different mitral cells. In addition to the animation, I would like to show you Blender user interface and how the program runs in Blender. The user interface is modified so that users can access the data in a visual way and can manage the visualization process. All soma are rendered by default so that users can select somas to render the other parts of mitrals by interactively right-clicking on somas. ID of the selected object can be looked through this panel. ID of soma set as ID of mitral cell. Another panel that shows the IDs of all objects currently rendered is here. If you want to render a specific mitral cell, you should first select the soma of the mitral cell by right-clicking on it, then go to the mitral cell panel from here and click Select Mitrals button, and when you finish your selection, you can click the Render button. Otherwise, you can continue to select other mitrals. You don't need to select mitrals one by one and click Select button every time. Group selection can be made by pressing B, then click Select Mitrals and Render buttons. If you didn't decide the mitrals you want to render, you can simply play the animation to get more information about data. After you decide which mitals you will render, you can follow the same procedure. As an example, I am selecting this mital and this mital is currently active in this region. As you see, tufts, epic, soma and then there is alt coloring. Another feature that is added to user interface is a curve that shows the number of active mitrals by a frame ID. If you look carefully, there are many active somas in this frame interval, as you see, and on the other hand, there is no active mitrals if you go to the other frame. This tool helps user in selection of mitral cells and time intervals interactively. The visualization tool doesn't only make the visualization of olfactory VAP data, but also allow users to analyze the data in a visual way. Different patterns on mitral cells by different odor inputs can be extracted with the help of this tool in an interactive way.